So yeah, my name's Minogia. I'm the lab manager of the Brosi Lab at the University of Washington and also currently the coordinator of MeadoWatch, which is a citizen science program directed by Dr. Barry Brosi and Dr. Yannicka Hill-Rees-Lambers, monitoring wildflower phenology at Mount Rainier. Next slide, please. So today I'm just going to tell you a little bit about what phenology is, what it is we do in the MeadoWatch program, how we do it, and then just a little bit about our plans for the future with pollinators. Next slide. So I mentioned that the goal of MeadoWatch is to monitor phenology. So what does that mean? Phenology basically refers to the life stage timing of organisms. So for example, the timing that birds migrate, when the leaves change color in trees, when bears hibernate, or when frogs lay their eggs. Next slide, please. So we're personally interested in the phenology of flowering plants. So that is the timing of when and for how long these plants undergo certain life stages. And that could be at the level of the individual plant or at the species level, thinking about the total duration of any individual of the species in bloom, for example. Next slide. So there are a variety of environmental cues that organisms use to determine their phenology, and many of them are related to climate, for example, temperature. And as you can imagine, phenology and shifts in phenology are strongly tied to climate and shifts in climate. So in our system, the subalpine meadows of Mount Rainier National Park and many other montane systems, plants often rely specifically on the timing of snowmelt as a phenological cue. For example, these beautiful glacier lilies here, Erythronium montanum, they're one of the first flowers to appear each season, and they're heavily reliant on snowmelt as a phenological cue. And we also know that while many of our plants use snowmelt as a cue, the sensitivity of different species to changes in snowmelt could differ greatly. Next slide, please. So studying phenology is becoming increasingly important because the climate is changing. And we're already starting to see some of those effects, like increasing average temperatures and melting glaciers. So spring events like snowmelt are expected to advance with climate change, and it's crucial that we understand how this is going to impact wildflower communities. Next slide. Next slide, please. Yeah, thanks. Phenological shifts spurred by earlier snowmelt could lead to several potential negative consequences. For example, there may be lower abundance and survival of these wildflowers, particularly for early season blooms that are more susceptible to events like frost damage in those unpredictable first days of spring. Additionally, because we don't expect every species to shift their phenology in the same way, plant interactions might change dramatically. For example, competitive dynamics between different wildflower species might change if plants that have never bloomed together are suddenly competing in these future climate scenarios. And then interactions with many other key organisms might be compromised as well. And that's something I'm going to get into a little bit more later on. Next slide. So with that, let me tell you a bit about our program and what we are doing to help answer these questions. So MeadoWatch is a long-term citizen science project monitoring wildflower phenology. This program was initiated over 10 years ago now in 2013 by Dr. Yannicka Hill-Rees-Lambers, and it's currently run by the Brosi Lab, and we work very closely with the National Park Service as well. Next slide. So MeadoWatch uses a citizen science approach, which is essentially the participation of the general public in conducting scientific research. And there are a lot of benefits to this. It allows us to collect data at a much larger scale than we possibly could with just a small team of researchers. That's especially great for studying processes like flowering, where there's a really quick turnaround time, and we really need to sample quite frequently to get a good idea of when exactly these phases are happening. It's also an incredible way to increase public engagement with science and research, and we're really grateful to have a dedicated set of volunteers who donate their time and effort to helping us collect this data. Some of our volunteers have been with the program since its inception 10 years ago, which is kind of incredible to think about. Next slide, please. So we're monitoring the phenology of these 16 focal wildflower species at the park. Here's just a couple of pictures of them. Next slide. We divide their life cycles into 4 key identifiable stages or phenophases, seen right here in a few example species. So we have budding, flowering, fruiting, and seed release stages. Next slide. So our volunteers record phenology at these 2 trails. Both trails follow an elevational gradient, as you can see in this image here, just with colors representing an increase in elevation. And that determines the timing and the duration of the growing season and also affects which species we actually see that are present. Next slide, please. So volunteers first undergo orientation and pass a quiz to become eligible for data collection, and then they follow instructions to locate our plots, which are marked by these small orange markers. We then use arm span to kind of mark out the area of the plot, and then we identify any focal species that are present in the plot, as well as which phenophases we see. And then we record that on a data sheet, which, next slide, looks something like this. So you can see volunteers record the phenophases, and sometimes there are multiple phenophases present in one plot, and then also record signs of herbivory and presence of snow. Next slide, please. And then at the end of the season, we go out and we deploy temperature loggers to determine the exact microclimate and timing of snowmelt at each of these individual plots. And here's just an example plot of a logger with time along the x-axis and temperature along the y. And you can kind of see exactly when the snow melted at this site right here, marked by that red arrow. So with all of this data, we can identify when exactly a focal species is in each phase in a certain year, and then we can compare that to what we know about that year's climate, both at the smaller plot level and at the park level. Next slide, please. So we've done quite a bit with this data, but I'll just quickly highlight an interesting result from a 2021 paper. So this graph right here, the left plot is Glacier Basin and the right is Reflection Lakes Trail, and you can see at both trails how earlier snowmelt years, which are represented by the lightly colored dots, are correlated with earlier peak wildflower bloom times. And then pretty interesting, this study also considered not just the correlation between snowmelt timing and peak flowering, but also snowmelt timing and visitors to the park. Next slide. Just to see if there could be a sort of social ecological mismatch. And the second graph here shows that is a possibility, with visitation to the park really remaining quite similar across years, regardless of when snowmelt occurred and subsequently when flowers were at peak bloom. So as climate changes continue to maybe shift flowering phenology, visitors are less likely to be changing their travel plans, and we may see a decrease in visitor satisfaction for those coming to the park to see these wildflower displays. Next slide, please. So I'm going to switch gears a bit now and tell you about some of our interesting ideas for the future of the program concerning pollinators. It's a super important relationship that directly dictates the success of these flowers. As I mentioned earlier, we expect that the phenology of many organisms is going to shift with climate change, including pollinators. And pollinator phenology can refer to many things. But we typically think about it as the timing of their foraging season. So maybe the day they first emerge and then the duration of when they're out pollinating. And we expect that plants and pollinators might use different cues for their phenology. Next slide, please. So what happens if plant phenology and insect or pollinator phenology rely on different environmental cues? For example, a pollinator that uses a cue like day length and its floral partner that relies on snowmelt might be in trouble because one of those cues is expected to shift with climate and the other isn't. And even if there are species of plants and pollinators using the same phenological cue, for example, both relying on snowmelt, it's unlikely that they have identical sensitivities. So in other words, it's unlikely they're going to shift their phenologies at the exact same rate. And that creates the potential for a plant pollinator mismatch in which there's no longer an overlap in the timing of interactions that usually occur. And that could be really detrimental to the successful pollination and reproduction of these plants, and also to the population of pollinators who rely on these plants for essential resources like pollen and nectar. Next slide, please. So we're proposing an expansion of MeadoWatch that would incorporate the citizen collection of pollinator visitation data, and that would include collecting observational data on the pollinators that visit our focal species, and then recording the identity of the pollinator, the plant species it visited, and then the time the interaction occurred. Next slide, please. So we're hoping this data will allow us to better understand whether a plant-pollinator mismatch is occurring and whether it could be a threat. We're also hoping to find out which plants interact with which pollinators and then be able to predict which pollinators are at risk based on the plant species we know are experiencing great phenological shifts. All that while hopefully increasing public engagement with pollinators. Next slide. And then I'll just end by saying anyone can be a MeadoWatch volunteer, and we're always super excited to get new volunteers. And so if you'd like more information on the process of becoming a volunteer this season, you can use this QR code to join our email listserv or visit the MeadoWatch website to learn a bit more about our research and also about becoming a volunteer. And yeah, be able to hike and camp in the park for free and contribute to some interesting research. Next slide, please. And here are all the people and the places that make this program possible. So huge shout out to all of these folks. And with that, I'm happy to take any questions.
Thank you so much, Minogia. That's a really exciting summary of all the amazing work so far. And that is interesting to think about including pollinators in that. Does anyone have any questions? We have a few minutes. I see a hand up. If you want to unmute yourself and ask, please go ahead. See, and there is a question in the chat. With the changing climate resulting in warmer temperatures, do you believe that eventually they'll reach normal levels again? And William, maybe I'm not understanding, but maybe it's thinking about, will they sort of equilibrate into a peak bloom again? Is that your thought, William? Yeah.
Yeah.
Do you have any thoughts on that, Minogia?
So I guess if I'm interpreting the question right, it's like, do we expect that with the climate warming, plants are going to shift their phenology, and then insects will also shift their phenology in the same way? And I think it's unlikely that they will become more synchronous with climate change. I think it's possible that insects might decide to shift their floral partners and their preferences for which species they're visiting, but it's unlikely that we would see the exact same interactions.
I think we have time for one more question. Oh, there's a question. This is not for Minogia. There's one about reservation and permitting, and we don't expect the reservation systems at the park to impact research at all.
Yes, and that's actually something that we double checked as well and can confirm. So that's definitely another bonus of being a volunteer.
Yeah. And the pollinators don't have to make any reservations at all. Well, thank you so much for your talk. This is really exciting, and I'm so excited to see what MeadoWatch does this year in the park.
Yeah, thank you.